Good morning, class. In our previous class, we had started with molecular basis of inheritance, in which we had discussed that how DNA has been proved to be the genetic material. Deoxyribonucleic acid is the genetic material which has been proved by many scientists like Griffith and Hershey and Chase in their experiments. Today we are going to do the structure of DNA, and we saw that DNA is the genetic material in most of the organisms, whereas in a few plant viruses and retroviruses like HIV, human immunodeficiency virus, RNA is the genetic material. So we will do the structure of DNA, structure of RNA, and difference between RNA and DNA today. Now coming back to DNA. DNA stands for deoxyribonucleic acid. It is a macromolecule, which means a single large molecule which is a polymer of nucleotides. And the nucleotides in DNA are deoxyribonucleotides, which is actually composed up of three components: a sugar, a base, and a sugar and a base together form a nucleoside, and then a phosphate group attaches to it. So the third component of a nucleotide is the phosphate group, and that's how sugar plus base plus phosphate forms a nucleotide, and several repeating nucleotides make up a single strand of DNA. DNA is a double-stranded helical molecule. It is made up of two strands. The two strands, each strand is made up of several polynucleotides, and each strand is a polynucleotide strand that is made up of several nucleotides. Each unit is a nucleotide. These two strands are coiled around each other like a helix, and these two strands are anti-parallel to each other. That is, if one runs in the 3-5- direction, the other strand runs in the 5-3- direction. We will be discussing the structure of DNA, which was proposed by Watson and Crick, and all these things will then become clear to you. Now before I move on, let us summarize what I have told you just now. So this is molecular basis of inheritance. The chapter is titled Molecular Basis of Inheritance, and we have proved that DNA, that is deoxyribonucleic acid, is DNA. Deoxyribonucleic acid is the genetic material in most of the organisms. Now, DNA is a macromolecule. Macro means large molecule. A single large molecule which is made up of several nucleotides. Or we also say it is a polymer of nucleotides. And these nucleotides are also called deoxyribonucleotides, and each is made up of three components. First, a sugar. This sugar is deoxyribose sugar, a 5-carbon sugar. Deoxyribose sugar, which is a 5-carbon sugar. The structure is like this. 5 carbons form a ring. This is first, second, third, fourth and fifth carbon. So first carbon and the second carbon lacks an oxygen. That is why we call it deoxyribose sugar. This is the structure of deoxyribose sugar. Second component is a base. Now bases are of two kinds, purines and pyrimidines. The purine bases are adenine and guanine, and the pyrimidine bases are thymine and cytosine. The third pyrimidine base is uracil, which is only found in RNA. In place of thymine, uracil is found in RNA. And the other kind of sugar is ribose sugar, which is found in RNA. And the third component is a phosphate group, or we can say phosphoric acid. And if we show the structure of phosphoric acid, we can say this is phosphoric acid. Now these are the three components of deoxyribonucleotides. Purines are these purines are double ringed compounds. And the pyrimidines are single ringed heterocyclic compounds. And each nucleotide is depicted as a sugar which is attached to a base, and on the fifth carbon atom a phosphate is attached. A phosphate group is attached like this. Okay, I will say CH2OH and this is OH. So a molecule of water is removed and a phosphodiester bond is formed. This bond that is formed here is a phosphodiester bond, and this sugar phosphate, this forms a backbone of the chain of nucleotides or a single strand of DNA. So if we see the structure of DNA, one more thing that I would like to tell you here, that how nucleotides are formed. This is clear to you? Okay, I am erasing this. Now, when a sugar combines with a base, we say a nucleoside is formed. And when sugar plus base plus phosphate, that is in a nucleoside, if phosphate is added, then a nucleotide is formed. So if we take into consideration the base adenine, when it combines with sugar, adenosine, or we can say deoxy, because the sugar is deoxyribose, so we say deoxyadenosine is formed. This is the name of the nucleoside. And when this combines with a phosphate, we say deoxyadenosine monophosphate. is formed, and we abbreviate it as DAMP. Same way, if guanine, then deoxyguanosine is formed. When phosphate, then it is called DGMP, deoxyguanosine monophosphate. If thymine, the base is called thymidine. The nucleoside is called thymidine because thymine is only found in combination with deoxyribose sugar. In RNA with ribose sugar, in place of thymine, uracil base combines and it is called uridine. So this is TMP, that is thymidine monophosphate. This is the nucleotide, this is the nucleoside, and this is the base. And if cytosine is there, we call it deoxycytidine. And the nucleotide that is formed is deoxycytidine monophosphate. So this is how the nucleotides are formed. Now this is one nucleotide. This is the sugar. This is one nucleotide. That is sugar. This combines with base. This is the second carbon. This is the third carbon. This is the fourth carbon, and this is the fifth carbon of the sugar. This is sugar. This is base, and this is phosphate group. So one molecule of water is removed and a phosphodiester bond is formed. So this is the formation of one nucleotide. If the base is adenine, we say DAMP, deoxyadenosine monophosphate is formed. If the base is guanine, we say DGMP, deoxyguanosine monophosphate is formed. If the base is thymine, then TMP, that is thymidine monophosphate is formed. If the base is cytosine, then DCMP, deoxycytidine monophosphate is formed. So this is the structure of one nucleotide, and several such nucleotides are repeated. Now this phosphodiester bond, make note of this. This phosphodiester bond is formed between a phosphate group with the fifth carbon atom of the sugar. This is the fifth carbon atom of the sugar, and this is the phosphate, and this is the third carbon atom. So more nucleotides are added like this. Now here, let us say another phosphate group. This is OH, double bond O, OH. I think I will have to erase it to show the whole structure to you. It is not fitting in here. So this you have understood. I am erasing this to show you the structure, to make you understand how a strand of DNA is formed. Now see, this is one sugar. This is carbon 1, carbon 2, carbon 3, carbon 4 and carbon 5. This is the fifth carbon atom. This is the first carbon atom. So with this a base attaches, the first carbon atom. This is the second carbon atom. In deoxyribose sugar, oxygen is missing. This is the third carbon atom. Now a phosphate group here. This is a phosphate group. This is a phosphate group. is one phosphate. This is double bond. Now, a molecule of water is removed. Okay. Now, the next nucleotide. The next nucleotide is this is the phosphate group. Which attaches to the fifth carbon atom of the sugar like this. Another base attaches here. So now a molecule of water is removed from here. So this is one nucleotide. Let us say this is one nucleotide. This is the second nucleotide. This is the second nucleotide. This is the so a phosphodiester bond. This is a phosphodiester bond. Oh, sorry. This is a phosphodiester bond, and this is the other phosphodiester bond that is going to be formed here. These are phosphodiester bonds by which several nucleotides are attached. The other chain runs anti-parallel like this. The bases are towards the inside, and there is hydrogen bond that is formed between the bases. The next base is like this, and this is carbon 1, carbon 2, carbon 3, carbon 4. This is the fifth carbon. So third and fifth carbon, they are united like this by a phosphodiester bond. Okay, and this is the fifth carbon atom. So the chain is like this between fifth carbon atom of the sugar and the third carbon atom of the next sugar. Third carbon atom and fifth carbon atom. This is fifth carbon atom, this is third carbon atom. So, and here this is fifth and this is third. So these are anti-parallel. One runs in the 5' to 3' direction, the other runs in the 3' to 5'. The bases are pointed towards the inside, and the sugar phosphate forms the skeleton of the DNA. And Watson and Crick said they are complementary to each other because adenine will always pair with thymine and cytosine will always pair with guanine and vice versa. Two weak hydrogen bonds are formed between adenine and thymine, and three weak hydrogen bonds are formed between cytosine and guanine. As I told you, one purine, adenine is a purine. Okay, let me write like this. Adenine and guanine are purines. Thymine and cytosine are pyrimidines. So one purine forms a bond with one pyrimidine, and the pairing is always between adenine and thymine and vice versa with two weak hydrogen bonds. Same way, purine guanine forms a bond with cytosine, three weak hydrogen bonds, and vice versa. This is Chargaff's rule, that always purine will pair with pyrimidine. That is why the amount of purine is always equal to the amount of pyrimidine. That is, we can say adenine plus guanine is always equal to cytosine plus thymine. And this ratio, adenine plus guanine upon cytosine plus thymine, is constant for a particular species. This ratio is constant. And the two are purine and pyrimidine are equal to each other. So this is Chargaff's rule. And the model for the structure of DNA was proposed by Watson, Crick and Wilkins, and it's called the Watson and Crick model of DNA, which says DNA is composed up of two polynucleotide chains forming a double helical or double helix. The two strands are complementary to each other because the bases are complementary, and the two strands run in opposite direction, are anti-parallel to each other. One is in the 5' to 3' direction and the other is in the 3' to 5' direction. I will tell you more points about the model, but this is the basic. Now coming to the salient features of Watson and Crick's model for DNA. It says DNA molecule consists of two polynucleotide chains, or we call them strands of DNA. These chains form a double helix like a spiral staircase. That is, when we say this double helix, erasing this, it means if one strand is like this, the other strand is like this. And this backbone is sugar phosphate backbone, and inside are the bases. Same way, sugar and the bases are inclined inwards, and this sugar and base, there is weak hydrogen bond between the bases. As I told you here, adenine and thymine have two weak hydrogen bonds, and cytosine and guanine have three hydrogen bonds between them. The two chains are anti-parallel. That is, if one is in the 5' to 3' direction, the other is in the 3' to 5' direction. One is 5' to 3' direction and the other is 3' to 5' direction. This is meant by anti-parallel chains. This is the polarity of the chains. The sugar phosphate units form the backbone and the nitrogenous bases form the center. Both the strands are joined together by weak hydrogen bonds which are present between the nitrogenous bases of the two strands. Pairing between the nitrogen bases is adenine pairs with thymine, cytosine pairs with guanine. This is Chargaff's rule, and such a base pairing is complementary. The base pairing is always between a purine and a pyrimidine. The double helix takes a complete turn after 3.4 nanometer, and 1 nanometer is 10 to the power minus 9 meters. There are 10 base pairs in one complete turn. When one complete turn takes, in between there are 10 base pairs. This is one complete turn. From here till here, there are 10 base pairs. There are 10 base pairs between them. And the distance between these 10 base pairs is 3.4 nanometer. So between one base pair, the distance is 0.34 nanometer. There are 10 base pairs in a complete turn, and the width, this is the width of DNA, which is 20 nanometer. The width of a DNA molecule is 0.20 nanometer. The nucleotides in a helix are joined together by phosphodiester bonds. Then Chargaff's base pairing rule, it says adenine always pairs with thymine and vice versa. Guanine always pairs with cytosine and vice versa. The amount of adenine is always equal to thymine in a DNA molecule. The amount of guanine is always equal to cytosine, and the amount of purine is equal to the amount of pyrimidine. Purine is adenine plus guanine. So adenine plus guanine is equal to pyrimidine, that is cytosine plus thymine. And the ratio of adenine plus thymine to cytosine plus guanine is constant for a particular species. All these points I have told you. Now we come to the structure of RNA. As I told you, in RNA is a single-stranded molecule. Unlike DNA, it is not a double helix molecule. RNA stands for ribonucleic acid. It is a single-stranded molecule. It is also made up of, it is a polymer of nucleotides, and the nucleotide in case of RNA is called ribonucleotide. And each ribonucleotide is composed up of a sugar. The sugar is ribose sugar. One sugar is composed up of a sugar, has three components, just like deoxyribonucleotides, three components, one sugar. Second is a base. The base can be either purine or the base can be pyrimidine. In case of RNA, the purine is adenine and guanine, whereas the pyrimidine is uracil and cytosine. Thymine is not present in RNA. And the third is a phosphate group or phosphoric acid. So same way, a sugar and a base. A sugar and a base form a nucleoside, and when a phosphate attaches to it, then it is called a nucleotide. So adenine, sugar plus adenine plus base forms adenosine. So the nucleotide formed with adenine will be adenosine. Monophosphate, or we abbreviate it as AMP. Formed with guanine will be guanosine monophosphate. With uracil will be uridine monophosphate, and with cytosine it will be cytidine monophosphate. So these are the names given to the ribonucleotides in RNA. So where is RNA found? RNA is present in nucleus and in cytoplasm. It plays an important role in protein synthesis and in the formation of ribosomes. Structure of RNA, it consists of a single polynucleotide chain, that is, it exists as a single strand. The polynucleotide chain is formed up of ribonucleotides. Ribonucleotides are also formed up of three components: a sugar, which is a ribose sugar, nitrogenous base can be purine or pyrimidine. Purine is adenine and guanine, and pyrimidine are uracil and cytosine. And a phosphate group, the nucleotides formed are adenosine monophosphate, AMP, guanosine monophosphate, GMP, cytidine monophosphate, CMP, uridine monophosphate, UMP. RNA does not follow Chargaff's rule. Important point to be noted. Like DNA, RNA does not follow Chargaff's rule. Its guanine contents are not necessarily equal to cytosine contents, and its adenine contents are not equal to uracil contents. Various ribonucleotides are also joined by phosphodiester bonds to form the polynucleotide chain. The chain of RNA strand has well-defined ends. One end is 3' polarity and the other end is of 5' polarity. You know, RNA helps in protein synthesis, and there are three kinds of RNA. RNA found in the ribosomes is called ribosomal RNA. RNA which helps in protein synthesis, that is when DNA is going to decide what protein is going to be formed, DNA first transcribes into messenger RNA, and then messenger RNA is changes into protein. The process of messenger RNA formation from DNA is called transcription, and from messenger RNA protein synthesis is called translation. And the amino acids that are brought for synthesis of protein are carried by transfer RNA. So there are three kinds of RNA: ribosomal RNA, messenger RNA, and transfer RNA, and we abbreviate them as ribosomal RNA is written as small r RNA, messenger RNA is written as small m RNA, and transfer RNA is written as tRNA. I will tell you the structure of these also, characteristics of these also, but basic structure of RNA I have explained to you. Now we will do the difference between DNA and RNA. Difference between DNA and RNA. DNA, deoxyribonucleic acid, RNA, ribonucleic acid. So we will write the differences between the two. So first difference, you know, the sugar present in DNA is deoxyribose sugar, whereas sugar present in RNA is ribose sugar. Then this is the genetic material in most of the cases, and RNA is not the genetic material except in a few cases like retroviruses and certain plant viruses. I am writing the differences in short. DNA is a double-stranded molecule made up of 2 strands. This is RNA is a single-stranded molecule. Then DNA, the molecular weight of DNA is very high, whereas the molecular weight of RNA is comparatively low. Then the length of DNA is very long, whereas RNA is shorter as compared to DNA. RNA is shorter. Then 2 types of DNA are there. One is intracellular DNA and the other is extracellular DNA. Two types, intracellular and extracellular DNA. And RNA is of several types. It is of many types. I have already told you it is of many types, but the 3 important types of RNA are ribosomal RNA, messenger RNA, and transfer RNA. Then DNA follows Chargaff's rules. Chargaff's rule, that is purine is always equal to pyrimidine, and adenine pairs with thymine and cytosine pairs with guanosine. Here it does not follow, RNA does not follow Chargaff's rule. Then the next difference is nitrogenous bases are adenine, guanine, cytosine and thymine. The nitrogenous bases are adenine, guanine, cytosine, and in place of thymine, uracil is present. U stands for uracil. Then DNA transcribes genetic information to RNA. And first, this is the central dogma principle. DNA first replicates. This cannot replicate. RNA cannot replicate. Then DNA transcribes. I told you formation of RNA from DNA is transcription. So it transcribes RNA. Whereas here from RNA protein is formed and the process is called translation. So RNA translates. It does not transcribe, it translates. And it converts the transcribed message of RNA to form proteins. Or we can even say polypeptide chains, several peptides made up of amino acids. So RNA forms protein. DNA controls genetics and metabolism, and RNA controls only metabolism, not responsible for transfer of genetic material, because DNA forms the genes. And we see that there is, because it is double helix, so we see there is twisting present in DNA, whereas there is no twisting present in RNA. So these are the major differences between deoxyribonucleic acid and ribonucleic acid. So we have discussed the structure of ribonucleic acid and deoxyribonucleic acid. We have discussed Chargaff's base pair ruling. We have discussed the basic structure of RNA and the differences between DNA and RNA. This much is enough for today. We will continue in the next class. Good luck till then.